Hello and welcome to Science Cafe Concord. My name is Dan Marchek and I'm one of the co-founders of Science Cafe New Hampshire. We're pleased to see such a good crowd here tonight. How many of you have not been to a Science Cafe before? Wow, that's a pretty good crowd. All right, so for those of you, Science Cafe is basically a question and answer. So our goal is to have the conversation reflect the questions that you have. So we will give the panelists a couple minutes to introduce themselves, and then we'll turn it over largely to you to tell us what it is that you want to know about.
Today's session is about a question already. Oh, wow.
I have no background in this area, but I know it's important. How about a 15-minute lead-in?
My first question will be, genetics 101. Absolutely, no question. So that actually leads to, that's a good point though, because this is being filmed, and this will be on Concord Television, and he will also, Sean will also have it on the website. He does an edited version as well as the full-length version, so you can, show it to your friends or relive the conversation later. But as a result, when you're going to ask a question, please raise your hand, and one of us will come over with a microphone. Don't just start talking out loud because the TV can't hear it. So, all right. With that in mind, today's topic is, as you all know, CRISPR and the breakthrough in sort of genetic modification technology and what that will and won't mean. And we have two panelists, both of whom are volunteering their time. I much appreciate it, from UNH. And you are both plant geneticists, is that correct? And Tom Davis and Subhash Minocha? Yeah, close enough. Yeah, I missed it close enough. So what I'm going to do is I'm going to give each one of them about 2 or 3 minutes to just say who they are and what the background is and why we should pay any attention to them, and then I will plunge into my first question, which, as you said, is going to be very basic, and then we will go into your questions from there.
All right. My name is Tom Davis. I'm a professor of plant biology and genetics at UNH. I've been at UNH now for a little over 30 years. I grew up in California. My wife Liz is here as well, and she's from California. And I was thinking about that, the fact that the first class that I taught at UNH, it was actually a plant biotechnology class. And one of the first questions I asked the class was, well, how do you feel about genetic engineering? This was in 1984. I said, how do you feel about genetic engineering? And one of the students said, he's from California, all right. He's asking us how we feel about something, So a lot has changed since then in the area of this topic. I got my bachelor's degree in biology at Cal Poly San Luis Obispo, and got my PhD in genetics at University of California, Davis. My interests are really focused in the changes that domesticated plants have undergone, the evolutionary changes that domesticated plants have undergone, all throughout really their evolutionary history, and that goes back to the beginning of life on Earth, if you really think about it. But the last 10,000 years is really the history of domesticated plants, and then the last 100 years or so is the history of genetics, and so I'm very interested in those topics, and I teach a lot of courses about them. My research is focused on developing genomic tools, sequence data, and tools of various kinds to apply to a process called marker-assisted breeding. And marker-assisted breeding is not genetic engineering. It's a form of breeding that utilizes DNA fingerprint type of information to try to recognize the genes that plants have and to select superior plants in a breeding program based not only on their obvious characteristics but on their genes as well. So this marker-assisted breeding, in any case, we're developing tools particularly for strawberry. I've been working in strawberry for quite a number of years. Strawberry is one of the most complex in terms of its genome, one of the most complex of all cultivated plants, and I can tell you a little bit about that later if it comes up. Just in the last couple of years, we've gotten into a project on looking at weed genomes, the genomes of weeds in the New England area, and whether we can determine which weeds might be likely to respond to climate change and evolve into new problems. And one of those weeds is called lamb's quarters, and that led us to get interested in a relative of lamb's quarters called quinoa. And so there's a very interesting story about domestication around that. And I have used some genetic engineering techniques in my research program, primarily to try to understand what some genes do. We're not using any form of GMO approach in our breeding program, and that's because until we feel comfortable that the public is ready for that, we want to breed strawberries and other plants that are suitable for organic production. And so we're staying away from any form of genetic engineering in relation to that, but it's something that I need to know about, and I'm really looking forward tonight to getting a sense from the audience here how you feel and what the kind of receptivity is now in relation to genetically modified crop plants. So that's one of the reasons I came tonight, is I'm really looking forward to learning and hearing what you all think.
I'm Subhash Minocha from the Department of Biological Sciences. I'm also a professor of plant biology and genetics. I came to UNH almost 42 years ago. So I'm finishing my 42nd year here. And I came from the University of Washington, Seattle, where I did my PhD in botany at that time. Like most other universities, they've all become biological sciences or biology now. But they still have a very strong program with plants. And there I was basically asking a very simple question to the plant cells, that how do plant cells become what they do, which is a whole plant? Because it was known that plant cells, we can give them orders, chemical orders, and they'll do almost anything you want them to do, become a root or a shoot or a stem or a leaf or a flower even, without going through the whole development of the plant. So that was a fundamental question that attracted me when I was in high school, and I kept up. Although if I had done what I started doing at Notre Dame, I spent a year there, I would be today producing these mosquitoes that breed with the natural mosquitoes and don't produce the next generation. That would have been my PhD project. So that tells you something about how life takes a change. Anyway, I spent a year at Notre Dame. I tried to get into their football team, couldn't get in, so moved to University of Washington and continued my botany. After I came here, myself and my wife and our family, we've lived in Germany and Norway and Japan and New Zealand for almost 8 months to 12 months on our sabbaticals. So I've always been interested in the education system of individual countries, compare that with the US, and been involved with quite a few things in the outreach. My research still kind of asks the same question. How do single cells know what they want to become, how we can manipulate them, how we can play with them, and most importantly, how can we understand that, how they do things, what they do? Since then, the stem cell people, which you I'm sure all have heard about, have taken on a highlight of the same type. How do animal cells or human cells know what they do and how we can manipulate them, take some cells and make them neurons, make them muscles, whatever we want. Separate issue completely. But that has involved an evolution of research in terms of asking a simple question of feed and study to genetically manipulate. So today I use the term metabolic engineering of the metabolic pathways in plants to understand how the pathways are regulated. And we've been working on a number of pathways, but the most important one of those involves carbon and nitrogen. And in more recent work, we have demonstrated that you could change nitrogen metabolism of the plant, and through that, you can draw in more carbon and increase the growth of plants, and we are testing that hypothesis with popular plants. We've done some work with model plants and with cell cultures and so on. So that's my research part. I also collaborate with my wife, who is a scientist in the USDA Forest Service in Durham itself. And her interest in developing biochemical markers of stress in forests. Ask the question like we do with humans. If your insulin levels are low, do you have a problem? If your blood sugar levels are high, you can tell that you have a problem at the individual level, but if you do epidemiology on a large scale, you can tell the population of Durham, New Hampshire, for example, is healthier than maybe the population of who knows what other town, I won't name any. You can do those studies. So you're looking for chemistry of the people, from that draw the conclusion that they're healthy or not. And there's a similar thing going on with trees. So my interest in fields like this is I teach courses in biotechnology for honors students, very freshman level class, and I teach upper level course in plant genetic engineering and biotechnology and similar to those.
Oh, we are not honor students here. We are the freshman class. So let's start out, as I said, with could one of you give, or both of you, give a 5-minute, 7-minute introduction into what is DNA and what are chromosomes and genes, these terms we sort of vaguely know, so we can now then understand when we talk about how they're being manipulated.
Little intro genetics, okay. When I was an undergraduate student, I was in a lab class where the project was to look at chromosomes. And you look at chromosomes under the microscope. And we did an exercise in which we looked at our own chromosomes. So we cultured white blood cells, which is the way to go about looking at chromosomes. And I told my mother about that, and I said, we looked at our own chromosomes, and she said, how did you do that? And I said, well, we put them on a microscope slide, and we looked at them under the microscope. And she said, but don't you need them? So what are they? I mean, it's you hear these terms, but what are they, and how many of them do you have, and so on. And really, that's where our genes are for the most part, are on these little objects, chromosomes, and we get a set of them from one parent and a set of them from our other parent. Unless you happen to be a strawberry, then you get 4 sets of chromosomes from each parent. You have 8 sets of chromosomes. And if Gregor Mendel had tried to discover the principles of heredity using the cultivated strawberry, we never would have heard of him. But ultimately, the chromosomes and the hereditary material, at the heart of it is a molecule, DNA, that can be isolated from living things and manipulated as a chemical. And that's what recombinant DNA technology is about. Thinking about what that molecule is. A chromosome, our bodies have trillions of cells, and each one of those cells has a nucleus, and in each one of those cells there's 2 sets of 23 chromosomes. And yet, in one of those tiny, minuscule little chromosomes, there's a DNA molecule that's about this long. It's just incredible. And then just millions and millions and millions of units along that molecule. And
you mean literally that long?
Literally this long.
And so it's all tied up and squished inside a cell?
It's all tied up and squished up into little bundles. And it's linear, and it's a linear molecule of subunits we call nucleotides. That's the A, T, C, and G. And it's really two linear molecules that are mirror images of each other and curled around each other in the double helix that we're familiar with. And it's the sequence of these 4 subunits, this A, T, C, and G, that carries all the information content. Well, now we know that's not really true because there's additional kinds of modifications to DNA that carry information as well. But the DNA molecule, its sequence of these 4 nucleotides, carries this tremendous amount of information. And that is what we would manipulate, or what would be manipulated in genetic engineering. Now, one thing is that all living organisms that we know of share the same genetic code, and that genetic code means sets of 3 of these nucleotides, A, T, C, and G, sets of 3 of them make up a code called a codon, and there are 64 of those codons in the DNA, and those codons have a correspondence to amino acids, and the DNA coding instructs the cell how to string together these amino acids into functional proteins. Well, all living things that we know of on Earth share the same genetic code, and so when I mentioned that when I studied the evolutionary history of strawberries, I'm going back to the origin of life on Earth, that's when that started, and that's why we can take genes from one organism, a bacterium, and put them in a plant or a human, and they can work, is because all these organisms trace back to a common ancestor, and we share a genetic code.
Okay.
So
Let me actually ask one more question before we get into here, because I think a lot of us have this idea that there's all these gazillion genes, and this gene, makes my nose too big, and this gene made my hair fall out, and this gene, made my eyes this color. But I'm sure it's not quite that simple as a one-to-one correspondence. So it's not like you can go in and snip away the gene that makes me, sleep poorly or something like that.
yeah, that's a great question. And for any particular characteristic of an organism, hundreds of genes interplay to influence that characteristic. But if you, and here's the important thing about genes, is that and in our genome there are about 30,000 genes. And each of those genes is made up of thousands of these nucleotide units. But genes come in different forms. Those forms are called alleles or allelomorphs, and that's a concept that goes all the way back to Gregor Mendel in 1865. And let's say you have a characteristic of an organism that involves hundreds or thousands of genes. Yet if you modify one of those genes or make it dysfunctional, that can have a tremendous effect on that trait. And so, for instance, there's many, many genes that influence how tall a pea plant grows, but if you knock out the function of just one of those, you can have a pea plant that only grows that tall. I'm going to hand the microphone over to Subhash.
Absolutely, absolutely. Is there anything else quickly you want to tell these freshmen before we go to the class Q&A?
Yeah, I would just put the same thing in a very simple, familiar way of looking at things. You have the Library of Congress, which has a lot of books, or library of any town has a lot of books. Each book has a lot of chapters. Take a book which is recipes for baking cakes. You have a bunch of things written. You can convert them into a DVD. Looks much smaller. That's what a chromosome is versus the library of books. You can convert that, put that on a flash drive, bring it up to the screen. Now you start playing with it. You start editing it. You start searching for recipes by keywords. Our cells do more or less the same thing. At all times, they're searching for what genes are going to be active, what genes should be turned off. And editing of genes, which will be very soon coming to through whatever methods, is basically look at that file on your computer and say, I want to change this sentence out of billions of sentences that are on the computer. Bill Gates has done a wonderful job that he says, choose the words by putting that somewhere on search, find the word, find the other word in between that. If you want to change, you can change it. That would be DNA sequence editing. The DNA sequence means everything that's on your computer under Microsoft Word. Some of it may be Excel, but that still follows very simple, similar principles of being able to be edited by you when you want it. And if you edit 1 or 2 words in the baking cake recipe, you may get a pizza.
All righty, let's
Yeah, go ahead.
let's go to questions here in the crowd. Don't be shy. Here we have a non-shy person.
Getting away from the technical nature of the question, Mr. Brooks in his article this weekend indicated, or today I guess, indicated that this gives us the ability of having the choice to give our daughters fluorescent hair, or curing disease. And as CRISPR provides a technology to do this very, very easily and quickly comparative to where we were 10 years ago, how quickly do you foresee the issues of ethics and moral value coming dragged into the whole question, and the potential of that delaying the technological progress that we can make?
I would say the question is already there. It has been discussed at certain levels, not at the level of public as much as it should be discussed. The question is already there, that this kind of a technology that allows you to edit the blueprint of what you are, what your children will be, is obviously a technology that is very simple, doable. We can do it at UNH, a hospital can do it in New Hampshire, and if something you can do at UNH, you can imagine how easy it is to do it. But doing it has consequences which are for what purpose? If you're doing it for the purpose of curing cystic fibrosis, I think most people agree, well, why not do it? We can cure a disease, genetic disease. If you are doing it to change the nature of the human being, because there's a certain part of that genome sequence that controls your behavior as well. If you're doing it for that purpose, then the ethics and the morals and the issues are endless. And we are right now excited about the technology. We are not sure, and that's, I think, part of the reason we are here to talk about. We're not sure if we should be jumping right into it or not.
Can I just
Wait,
add one point to that?
Let him, yep.
Yeah, I just add one point about having a daughter with fluorescent hair. Is you'd have to start from scratch on that. You couldn't use CRISPR to give your current daughter fluorescent hair. And these kinds of manipulations, they need to start with a single cell to generate an individual that is entirely genetically modified. And so if you, that single cell in an animal system would be a fertilized egg or an egg. In plants, you can get whole plants to grow up from single cells taken from a leaf or different parts of the plant. But in either case, there's a starting point of a single cell, and the time it would take to get from that single cell to an organism that would display the characteristic, it could be many years. And so in terms of, okay, what about human applications? Well, if we started today, we wouldn't know how it's going to work out for quite some time. You know, it would take a while to see if there was going to be some surprises there, which it seems like there usually are, right?
How can you be sure that what you do for this generation isn't going to have lasting effects for the next generation or the generation following that? I mean, are we creating an island of Dr. Moreau where maybe the consequences aren't what we're expecting?
Well, I mean, you can speculate about all sorts of possibilities. I think an important context for all of this is to understand that that nature and evolution operate, well, evolution has operated on the basis of mutations and genetic change. And so our genetic material is not totally 100% stable and perfectly passed on from one generation to the next as it is. We wouldn't be here today if it wasn't for the fact that the processes of DNA replicating and so on are quote unquote error prone. And so we are the products of mutations. And this CRISPR technology, looking back over the last 30 or 40 years of biotechnology, this CRISPR technology is bringing us closer and closer to the kinds of of genetic changes that are happening all the time in nature anyway. And so whether or not the change itself, at least some of the kinds of changes we might make with CRISPR, they actually will be indistinguishable from changes that could have occurred and that do occur in nature. And so it may not be reasonable to worry that somewhere down the line, future generations, it's going to blossom out into some monstrous thing. Now, that's not to say that all applications of the technology are equal, but at least some of them are going to be indistinguishable from what nature already does.
I'll just give a slightly different tone to the same. I think many of you are from the generation where green revolution made a difference to our food structures. And that green revolution happened mostly in 50s and 60s. I was a high school student when I first time visited the Indian Agriculture Research Institute, and I saw what they were doing. They had a little tower that went up and down, 30 feet into ground and 20 feet up. In that tower, they had about, I think of this size, a little globe that had cobalt-60, a few grams of it. Cobalt-60, if you have heard about this, is one of the most radioactive compounds, and they had to keep it 30 feet below ground so it doesn't cause any harm to surroundings. Around 11 o'clock, and I happened to be in the lab during that time, they'll turn on a siren and ask all the workers to leave that area. There'll be nothing around for about 200 yards around, and they'll raise that tower for about 30 to 40 minutes to irradiate all the rice and wheat crops they were growing. They'll do that every alternate day for about 2 weeks when the plants were flowering. After a few weeks, they'll go collect the seeds, grow them, go to the field, choose what they want, throw away what they don't want, breed standard techniques. What they were doing was creating mutations, changes. The difference between that time and CRISPR is very simple. You don't need cobalt-60. You can be very precise where you want the mutation without, so far at least whatever little knowledge we have, without worrying about side effects of that to other genes, because it is a very precise editing mechanism. If that contrast makes sense, then what we have been doing for the last hundreds of years, including Mendel telling us how it works, other than we've been doing even before that, and what we are doing today, the difference is the technology, a cell phone or an iPhone versus the good old telephone that was invented, the very first one where you sit there and try to talk. It's question of efficacy. On the other hand, efficacy is what has brought to the questions of morals, ethics, because now we can do it so fast. We can do it at UNH. We don't have to be at a big place. And that's, I think, the controversial part of this whole thing.
How do you know which genes will do what, and what changes to those genes, what effects those will have as a corollary?
Which genes do what is what we have been doing or studying for the last 50 to 65 years since we got to know a little bit more about the DNA structure. That's what we've been working on. Once we know which genes do what, now we can go back and edit those specifically. So it's like, as I said, Microsoft Word. Once you know what sentence is easy to get to, we can get to it and modify it rather than scramble everything and restart the whole book from scrambled sentences. So we are, I think right now the target would be mostly genes whose functions we think we know or we have evidence that we know.
All right, I think we're out of time. Please join me in thanking our panelists.